This is my demo of the glucose transporter system. On the left, we have the GLUT lumen. It contains sodium, glucose, and water. It is very high in concentrations of sodium. It is connected to the epithelial cell by the HGLT1 cotransporter, which is a carrier protein used in facilitated diffusion. Within the epithelial cell, we have potassium, water, and glucose. It is low in sodium concentration. Connected to the epithelial cell is the extracellular fluid by the sodium-potassium pump. In the extracellular fluid, we have sodium, water, and potassium. The glucose transport system starts within the GLUT lumen, where glucose is being transported into the epithelial cell using the concentration gradient of sodium between the GLUT lumen and the epithelial cell, because there's a higher concentration of sodium here and a lower in the epithelial cell. So using ATP, sodium and glucose are co-transported into the cell. Once the epithelial cell gets a high concentration of sodium, water actively transports into the cell as well because the cell has become hypertonic. The cell now needs to reduce its sodium concentration in order to keep the glucose transporting to continue. So it uses an active transport system called the sodium-potassium pump. Sodium enters the pump and binds with it, and then once again ATP is used and allows the pump to phosphorylate, changing its shape and allowing the sodium to enter the extracellular fluid. Now potassium is able to bind to the pump, and once again the pump changes its shape, causing the phosphate to leave and the potassium to enter the cell, regulating the concentration and allowing for the whole process to repeat again.